Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy of my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius podcast, now part of the Finding Genius Foundation. I have a quick note about the foundation. We're working on our anxiety and depression massive literature review. The goal here is to go through thousands of different sources, peer-reviewed papers, books, lectures, interviews, etc., put it all together into a low or no-cost resource for people that suffer from anxiety or depression, and put that on offer, to help people. The goal here is to try to find every treatment possible, and again, put it all into one curated place. So if you have interest in looking into that, go to findinggeniusfoundation.org. And today, my guest is Nancy Moran. She's the Warren J. and Viola Mae Raymer Chair, Professor of Integrative Biology, all at UT Austin, University of Texas at Austin. So we're going to talk about her work. So Nancy, thank you for coming.
Great. Yeah. Thanks for having me, Rich.
Yeah. Well, tell me, offline, we mentioned just a few, words about bees and aphids and different things. What's your research about currently? What are you working on?
Well, we still work on these sort of two main systems. And the thing that joins it, we're studying how bacteria or just microorganisms in general interact with insects. And we sort of use the insects really as kind of a system of just looking at how an animal interacts with bacteria especially, but microorganisms in general. So people have probably read about microbiomes and how microbes interact with humans, but really they interact with all kinds of organisms, including insects. And so our focus has been on that in insects, and specifically on these two very different kinds of insects, bees and aphids.
Yeah. So I guess maybe we'll start with bees. So where do they tend to pick up microbes? And, has anyone sequenced the bees microbiome to see what it looks like?
Yeah, so we started on this kind of project, I think it was 2007. People who have memories that long might remember that was the very beginning of this thing called colony collapse syndrome, where some bee colonies were suddenly dying that year. It was kind of an unexpected thing that happened, and the beekeepers, and no one really understood why. So there was some effort to look for the causes. And that's when I first got involved in this. And a major effort that was people at a number of different universities was to get a bunch of these samples of the bees, the sick bees and the healthy bees, and then to just do a lot of sequencing to see if maybe there's a new disease agent or something like that. Maybe a new disease agent, some kind of virus or bacterium had entered, the U.S. and gotten to our beehives. Well, it turned out that wasn't the case. There wasn't any one disease. There were a lot of different diseases, but there wasn't anything particularly new that year. But what happened was, out of that study, it became apparent that every honeybee colony and every single honeybee worker, so all the bees you see flying around, has within them kind of a really consistent set of bacteria, about 8 species of bacteria. And every bee has the same 8. And they make up almost everything in the bee. And those bacteria don't live anywhere outside the bee, right? So they're really kind of a bee thing, a little bee bacterial community that's sort of part of being a bee. And we got interested in that and kept working.
Yeah, what kind of bacteria have you observed in bee microbiomes? I'm sure there's probably crossover with people, but I'm sure there's bacteria that are radically different as well, right?
Mainly, there's no crossover with people. These are pretty much bacteria that live only in the honeybee in the gut. And so they're basically about 8 species. They were new species to science. So, we've spent the last decade, kind of doing just fundamental work, growing them and sequencing their genomes and, seeing what effects they have on the bees. But they're very distinctive to bees and specifically to honeybees. So they've kind of evolved with bees over a long period of time.
Well, I would think a bee's microbiome would change radically as it pollinates throughout the year. If they're doing almonds in a cold climate, let's say in Northern California, has anyone looked, before they pollinate almonds, after they do it? Blueberries before, blueberries after, et cetera, like throughout the year?
Pretty much, that's the striking thing. It really doesn't matter. Of course, they ingest different bacteria when they forage in different environments and stuff, but those bacteria don't live in the bee. They just kind of go through and, they're part of the food, really. So
I would think that the bacteria, I guess maybe they're multi-purpose. They can produce the same metabolites or short-chain fatty acids or whatever is needed for the bee, whether the bee is eating A, B, or C, right?
Right. And one thing to remember is bees always eat nectar and pollen, right? So even though those do differ a little bit between plants, there's a lot of similarities too. They have a pretty narrow diet when you think about it, pollen and nectar. And so almond pollen will differ from, some other pollen maybe, but mostly it's very similar. The same kinds of components are in there. And one thing we've discovered about, maybe one of the interesting things about what these bacteria do in the bee is it's really got a lot of parallels to the human microbiome. But these bacteria can break down the cell wall of the pollen. So traditionally, when people have looked at nutrition of bees, they thought, well, they use nectar, that's sugar, so that's their energy, and then pollen, that's the protein, because the pollen grain has protein in it. So there's energy and protein, and that's what the bees need. But it turns out that the cell wall, or the husk on the outer coat of the pollen, that's not protein, that's made up of these complex carbohydrates, basically fiber, we would call it, things like hemicellulose and pectin, those things that plants make. And there's a lot of energy stored in there, but it's very hard to digest. Usually animals can't, like insects or mammals, cannot digest those complex fiber polysaccharides. And so it turns out these bacteria they have enzymes that they secrete that break down those components so the bee can get energy from the pollen, from the husk of the pollen, not just from nectar.
What about the queen bee's microbiome versus workers versus, maybe it's the same thing, drones, the different types of bees in a hive? Has anyone looked, again, at their different microbiomes at the same time to see if they do differ?
Yeah, so that's a really interesting thing because if you compare workers to queens, and we've done that, a few people have done it, and very consistent results. It turns out workers, they have, like I said, this very consistent, 8 species of bacteria. They live in the certain places in the gut, and they're localized in a certain way. The queen is totally different. She doesn't have any of them, and she actually has a sort of a smaller microbiome, and really doesn't seem to have this dominant kind of consistent microbiome that the workers have. And so it's really interesting because, genetically, you probably know it, but the queen and the workers, they're the same. They're, it's only a developmental pathway that switches between them. Genetically, they're similar to each other. So there's something different about the queens. And it makes sense, I think, because the queen is an egg-laying machine, right? Her purpose is to lay eggs and to just keep laying them to increase the colony numbers. And the workers are the ones that deal with all the food and digestion and all the processing of food, which is what the microbiome is involved in.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit FindingGeniusPodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit FindingGeniusPodcast.com and click Support Us today. Now, back to the show.
Has anyone looked again across, if I have 2 hives, and each one has its own queen, and I sample them, do they differ amongst the hives as well?
It's pretty simple. Like there's these 8 species and every bee has the same 8 species. So at that level, it looks the same from one hive to the next. But within the species, I'm talking about the bacterial species, within the worker bee gut, within each of those species, there's strain variation. That's a lot more fine scale. So closely related strains, they might differ genetically a little bit, and that might affect the bee. So at that level of strains, there are probably some differences. It's a little harder to look at because you really have to sequence whole genomes to find these small differences. But at the species level, it's really consistent across individual bees and across hives around the world. I mean, that's sort of what, when we first looked at this a little in 2007, it was really striking. Bees from Europe, from Asia, from Australia, from the US, they all had those same 8 species and, very distinctive. And so that's really what got me interested in this to start with was, this seemed like something special about these particular set of species.
What about the metabolic pathways, or I guess the metagenomics of the 8 species? Has that been looked at, and do people understand what role these 8 species are playing? What metabolites they're producing in exchange for the food they get from the bee?
Yeah, there's been quite a bit. So the 8 species, they differ from one another. Some of them are involved in, like I mentioned, breaking down the components of the pollen cell wall. So there's a big complex molecule called pectin, which is in plant cell walls. And it has many, many different kinds of chemical bonds. And each one of those takes a different type of enzyme to break it down, right? So there's this whole set of enzymes that you need if you're gonna digest pectin. But ultimately, if you digest it, it produces all these sugars. And those have energy. Those are a source of energy, but they're kind of weird sugars like xyla. They're not like glucose and sucrose that we use in our diets. They're odd sugars like arabinose and xylose, so on. And so usually animals cannot use those odd sugars, but these bacteria can. They can ferment those sugars and they then can basically generate like short chain fatty acid sources of energy that can be absorbed by the bee. So that part of the metabolism is a really dominant feature in the microbiome. And then there's a lot of other things going on. They can, besides using these sugars, they can make all of the amino acids. So it's that the bee might need in its nutrition. So it's unclear, but possibly these bacteria actually provision amino acids that might be limiting in the bee diet because they have the pathways for making these. So we have sequenced many genomes and we can reconstruct the metabolism, for all of these species. The harder thing is seeing what do they really do in the bee, putting them back in the bee and looking at how do they really affect a living bee. So that's a hard kind of experiment, but we try to do those too.
When we were talking about the different types, I mentioned the queen and the workers, but what other types of bees are there in a colony?
Yeah,
so they have different microbiomes, the babies and the...
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Yeah, so the larvae, again, so the key thing about the honeybee microbiome is it's in the adult workers. So those are the ones that most people see flying around. They're in the nest. They make up all the bees that you see. But of course, their early life is spent as a larva inside the honeycomb. And at that stage, as a larva, they have a very different microbiome, very small microbiome. It's very different from that of the adults. They don't have any of the ones that the adults have. And so they really only acquire this more distinctive microbiome when they emerge as adults. So they go through the larval stages and they're being fed this very specialized food from the nurse bees. Then they pupate within the honeycomb there. and then they emerge, they come out of the cells, and then they're adults. And so on the very first day that they emerge as adults, they start to acquire the microbiome from the other workers, which are their sisters in the hive. So basically they get colonized immediately, within hours of coming out, they start to be colonized with the microbiome, and they get it through social interactions. Basically, they get it from other bees that have it.
Has anyone looked at, if there's anyone left in a colony after colony collapse disorder and sampled the microbiome of like maybe even the dead bees that are left, or are the microbes gone by then? What if a bee was attacked by varroa mites? Has anyone looked at the microbiome then to see, how it changes in the bee? Where the varroa mite itself, it probably has its own microbiome too.
Yeah, so there's not big changes. So I think one thing that happens is that if a worker is really stressed, say maybe it's starving or it's attacked by mites, varroa mites, or has a viral disease, then the microbiome will kind of start to go haywire, and it'll just look more basically contaminated with a lot of random bacteria from the environment. But that's usually a very brief stage before the bee's just dead, right? And so because the honeybees will just rather quickly die if they go into this very stressed state. So if we have like sick bees that maybe have nosema infections, that's another kind of parasite in the intestines of bees. They can have microbiomes that look very weird and that are starting to be kind of chaotic with kind of random bacteria. But we don't sample those very often, mainly because I think they just die very quickly and you don't really capture that moment very often.
Oh, but I was going to say, has anyone been able to, again, try to sample a bee or a couple of bees at a time when their colony, let's say, has collapsed or there's radical changes going on?
Yeah, we have. At the very beginning, we did that. And, they mostly will have the same microbiome for the most part. It'll just sometimes start to have other bacteria present. A lot of times there's kind of a collection of kind of pathogenic bacteria like Klebsiella and Serratia. Those are not normal components of the microbiome, but suddenly they'll flare up. Usually they're maybe present at tiny, tiny numbers, way less than 1%, but suddenly they'll be dominant. And so sometimes you can get bees in that state. One thing we've looked at is what happens when you treat bees, like bee colonies are exposed to different chemicals. So antibiotics are sometimes used in beekeeping, and another thing we've looked at is it's an herbicide, glyphosate. It's the most used herbicide in the world. And both of those have very strong impacts on the microbiota of bees because they're toxic to bacteria, both of them. And so they will really disrupt the communities. And so in agriculture, sometimes bees are exposed to these things, and those are situations where they will have, really disrupted microbiota composition, and it has effects on the bees.
Even after, let's say, the winter and the bees are hibernating, their microbiome is the same? Or I know that people that raise pollinator bees, they'll keep them in like fridges at 40 some odd degrees. And then they warm them up and they bring them back to life. But has anyone looked, during a cold period or after that, if the microbiome is the same?
There's been a little bit. We haven't done that very much, but that would be interesting to do. Like, we're here in Texas, and so pretty much it gets warm enough all winter for the bees to fly outside now and then, at least, because 55 degrees Fahrenheit and higher, they can fly around. And so one interesting thing about honeybee workers is they don't defecate in the hive. And so in cold places, they're sitting inside the hive all winter long, covered with snow, and they're holding it in. And the microbiome does shift a little bit. I mean, they have a very, full rectum, and, the whole gut physiology is affected. And there are some studies, there's sort of shifts in the proportions of species. They still mostly maintain a similar species set, but some of them might become more numerous and others decline a little bit.
Okay. So what fermentation do you want to do now to figure out more about bee microbiomes? Like what things do you have going on? What hypotheses or experiments are you working on?
Well, lately we've actually gotten kind of interested in actually practical things that might come out of this, things that might help bees. And one interest is, can we devise like a kind of a cocktail of healthy bacteria that might help help bees who might have been exposed to stress or different chemicals. It might help them get back to normal. Or like you said, after, like people who grow bees for pollination, after the bees have been shipped or subjected to stressful conditions, can we use these bacteria to help the bees, sort of become healthy and active again? So we've been playing around with just compositions of, sets of bacteria that we can give to the bees. And then a little farther out is actually engineering these bacteria. So we have engineered, that means changing them genetically. So genetic modification of the bacteria. We've taken advantage of a particular pathway that's in bees and other animals and plants that's called the RNA interference pathway. So it's basically we can give a given RNA molecule to the bee that will cause it to attack, say, a virus. So we've done this for the major virus that's in bees, the deformed wing virus. It's an RNA virus, and so we basically could engineer the bacteria to kind of, it's almost like a vaccine or stimulating the immune system of the bee to be ready to attack that virus. And it actually works. But so far we've only done it in the lab. So whether it's a practical thing outdoors is not yet clear.
Again, has anyone looked at the microbiome of the Varroa mites, Varroa destructor? Maybe there's a key there in one of the microbes that is in it, if there is. And
I don't think anyone has. Actually, we haven't, and I don't know anything on that. Some mites have very little microbiome. I mean, things that feed on fluids from other organisms sometimes have very minimal microbiomes in terms of at least the gut microbiome. But I'm not sure. I don't think anyone's looked at that. That would be worth doing.
Okay. You mentioned in the beginning of the interview, you also work with the aphids. Yeah. I would guess aphids and ants kind of are bedfellows, right? Where do aphids tend to be?
Yeah, some aphids, yeah. So, I mean, I've actually worked on aphids much longer than on bees. And again, though, I worked on bacteria that live in aphids. And aphids, bees are actually very smart insects. They can fly around, find their nest, do all kinds of complicated behaviors. Aphids are not as smart. They're kind of almost like the plant. But aphids contain a different kind of microbial association. And so they have these intracellular, very specialized bacteria. So every aphid has this one kind of bacterium called Buchnera. And it's just one. At the dawn of aphids, which is like 150 million years ago, so back when the dinosaurs roamed, basically they gained this endosymbiotic bacterium, and it has lived in them ever since, so 150 million years, and it's passed on when the aphids reproduce. So when the mother aphids give birth, their daughters get it, and so it's passed on through the maternal line. And it's just like mitochondria, if you know how those are passed on. So basically you get them from your mother, come with the egg, and they're passed on for millions of years and basically through the maternal line. And originally mitochondria were bacteria. So Buchnera is just kind of a more recent, it's very similar in many ways, but it's still more, its machinery is still more like a real bacterium. It still makes all its own proteins, and it has its genome is not is reduced, but not as far as mitochondria genome. So it's got a lot of parallels. It's evolved with the host. So we've studied that for a long time, trying to understand how it works, what it does in the aphids, and how it has evolved with them and has affected their evolution.
Okay, well, yeah, what are you trying to figure out in regards to the aphids' microbiomes? Do they change, or are they very static like the bees are?
Yeah, so aphids, they're interesting. They feed on phloem sap of plants. That's their exclusive diet. So they have these sort of syringe-like mouth parts. They inject it into the phloem cells of the plant, and then phloem's under positive pressure, so it comes through the aphid, and it's full of sugar and has amino acids and so on. And interestingly, things that feed on phloem sap or plant sap in general, they often have almost no gut microbiome. So their guts are pretty much sterile. It kind of makes sense because phloem sap is sterile, and it's a very clean thing to feed on. So you're not going to get many pathogens coming in through the diet when you feed on that. But so instead, they don't have a gut microbiome, but instead they have these intracellular bacteria that live inside special cells and that are part of the body. And most aphids, well, almost all aphids have this one called Buchnera. They all have this same one that came from a single ancestor one time. And of course, it's diversified along with aphids diversifying, but still it all came from one ancestor. And then sometimes they have one or two additional symbionts. And so what it looks like is this main bacterium, which is called the primary symbiont. Over time, it sort of loses genes. It sort of, because it is passed on from aphid to aphid and strictly clonal, it never can recombine with other bacteria. It loses genes and it never gets them back, right? So its genome is getting smaller and smaller and smaller over time. And so it's down to only about, some of them have as few as 400 genes, which is really close to your minimal, smallest cellular genome known. Right. And so it's losing things. And what we think happens is sometimes it's become so decrepit and lost so many genes that it benefits the aphid, the host, to gain a new symbiont to kind of take over some of the jobs that the old symbiont is getting really bad at. So it's sort of like senescence, it's getting worse and worse. Very specialized to serve the host, but it's kind of losing its capabilities.
Oh, one thing you brought up that came to mind earlier is, don't bees have a spermatheca? They could store sperm from, I forget what the males are called, for a long time.
Yeah, drones, yeah.
And has anyone looked in the spermatheca for microbes? Or, I know, like on the humans, they're not just in the gut; they're in many places. So I would think in bees, maybe they are as well.
So queens have the spermatheca, and they can store sperm from the drones for several years. And but I don't think anyone's looked at that for microbial, associates there. So I don't think that's been looked at. There's only been a few studies on queens. It's hard to get lots of queens, so people don't study them as much. But I don't think anyone's looked at that. Okay.
Yeah, I was just wondering, it might be an interesting place to look because of the the abilities of it to store things for a very long time.
Right. It is amazing that sperm can live queens can live 5 years and keep reproducing using the same little pot of sperm. So.
Yeah. Have you looked at microbiomes of ants? I guess, they culture aphids and they farm them and stuff and they hang out with them. So I guess it would make sense maybe to look at aphids and ants and see if they share. Yeah,
we haven't studied them, but some other people, one of my former students has studied ant microbiomes a lot. And so they, different ant groups really differ, and they never acquire the main symbiont of aphids. It's so specialized, it really stays in the aphid. It's like a mitochondrion. It's pretty much not going to get out of there. But like they do have microbiomes that are pretty interesting sometimes. And sometimes they also have kind of an intracellular symbiont that lives inside cells. And sometimes they just have a gut microbiome. And sometimes they have very specialized microbiomes that are involved in processing their food. So, ants have very different groups of ants have very different diets. Some of them use a lot of nectar. Some of them feed, on different sources. And so that seems to be linked to what kind of microbiome they have.
Well, very good. What do you see as the future of this research? What are some of the big questions that you and other people are working on?
Well, with the aphid symbiont work, one thing we're interested in is the symbiont is almost like an organelle. It's so specialized. It lives only within the cells. It cannot be grown outside the host is one thing I didn't mention. So it's only living inside there. And one thing we wonder is, as it evolves with the host, so you have a bacterium and then an animal host, are there sort of totally new things that evolve? And we've gotten interested in, are there new protein complexes that involve both proteins of the symbiont and proteins of the host that interact together to make, say, a new structure or a novel thing? So we know from mitochondria that within cell, like our cells or yeast cells, that the proteins that come from the mitochondria and from the host, they actually go together to form these sort of entities that require both genomes to contribute, right? So we're using the aphid system to kind of look at a younger version of that, so the aphid symbiont is evolved more recently. It's not as far down that road of just being part of the cell, but it's still very, very close and has evolved millions of years with the host. So there could be some interesting sort of novel biochemical, structures that have evolved from this.
Okay. Well, very good. Nancy, what's the best way for people to find out more about your work? Where can they go?
Well, let's see. I have a web page at the university with many publications on there. Some of them are written for a broad audience. That's University of Texas, Nancy Moran. If you Google it, you'll find the webpage and various things on there about aphids and their symbionts and a lot of things about bees and their bacteria. So I think those are probably good sources just listed on there.
Well, very good one, Nancy. Thank you for coming on the podcast. I appreciate it. It's been an interesting call.
Good. Yeah, thanks a lot. That was fun.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.